Hi class, happy Tuesday. Let's start out by completing the daily attendance for today. And then we will scroll down to this week's folder. And today is Tuesday. I expect you to be at your Google Meet for class. You're going to do your daily attendance form and the bell ringer. And we're going to start a virtual microscope lab. And I've placed it in this folder, but I'm going to have a link for it in your lab folder so you can find it your way to it both places. This lab will be due this Sunday by midnight. I'm giving you 2 class periods and a lab period to work on it. And then I also put in here that you're going to have a test on Friday. It's not going to be a long test, but it's going to cover the scientific method, lab procedures, metric system, microscope, graphing, pretty much everything that we've been learning so far. The test is going to be timed. And so once you open it, you only have that one session to complete it. You are not allowed to close it and then open it later. It will be found on Castle Learning. And I really think you should attend class, your Google Meet in class on Friday so that you can have all the instructions about how to take the test. So just so you know, the test will be available for a limited amount of time. You need to really make sure you get all your back work done before this test. After the test is completed, I will not accept any more late work. So that's something that you should know. Okay, let's go into our Tuesday folder. You have your bell ringer and then the virtual microscope lab. So here's what the lab will look like. It's going to take you 2 class periods and a lab to complete. Oh, you know what? I'm going to change this. It'll be due on Sunday, not Friday. I'm going to give you a little extra time. You're going to click the link to take you to the virtual microscope website. You're going to open this lab sheet using Cami, follow the directions on the lab sheet to complete the tasks. And then you're going to click the turn-in button within Cami. You don't submit it here. You're going to use the Cami submit button. So when we open it, let me go back to the top here. There we go. Okay, so this is your link to open the actual microscope portion. And so this is how it starts out. You can see your microscope is sitting here. And then what we'll do is we have to go down a little bit here. The directions on your sheet say you can use the Learn tab to familiarize yourself with the microscope and then click on Explore to look at your specimen. So the Learn, we're going to click that so that you can see what each of these things are. Okay, and it gives you a check mark so you've read them and you go through and look at everything. Once you've done that, you can go back to the main screen. And we'll go to the Explore. And this is where you're actually going to be completing your lab. So your sketches should be drawn as you see them in the field of view. You have different adjustments, and I'll show you how to use those. When you do your sketches, I recommend that you zoom in a lot, and I would recommend you do this on your Chromebook so you have your touch screen capabilities. I wouldn't use the mouse pad or a mouse if I were you, because you're going to be using your drawing tool and you're going to have to be drawing, little cells, and I think you have more control if you use your hand. So that's my recommendation to you. So here's what this is going to look like. You're going to open this. The first slide they wanted you to look at was the letter E. And so this shows the letter E. Here's all your different magnifications, and that's what these little objectives are. So at 4x, you can see this is blurry, but we have our coarse focus, fine focus, and the light that we can adjust. So you can take your coarse focus and move it around, and you can see when you put the letter E on the microscope the normal way so that you can read it, what happens is that it flips it upside down and reverses it. So you have to remember that no matter what sample you're looking at. And then you're going to draw this in your first circle to the best of your ability. I'm not grading you on artistic talent. I'm just grading on what you're seeing or what you should be seeing. And then you're going to go to the 10x. So this will enlarge it a bit more, kind of brings you closer, and you can play around with your coarse adjustment or the fine adjustment if you think you need to. If you think you need more light, you can do that. You're going to draw it exactly at what you see in that field. And then we're going to go to the 40x. And you're going to draw exactly what you see here. You can, make it better focus, more light. And so we keep enlarging and getting closer and closer. And then the 100 is even more on here. So we're going to click on the 100. But because this is the highest magnification, this is our oil immersion lens. So now we need to actually use oil that gives it a nice, crisp, clear image. So we're going to use this question mark up here. We're adding oil, and then we can use light to adjust it and the fine focus. And you can see how we're very much enlarged on here, 100 times bigger than what it actually is. So you're going to finish drawing that in your 100x, and we'll go out a little bit here, and then you're going to answer some questions about what you had to do, and then you'll move on to plant cells. So we'll go back in, we remove the slide, we have to clean with lens paper, which is right here. It's going to wipe off the oil, put the slide away, and we're going to go in here and get our plant cells. Take a look, and you can see it's blurry, so we're going to go through and do the coarse focus. And all these little brick shapes are plant cells. So I'm going to move it so I'm not in that thick part. And then you're going to go and you're going to draw all the little tiny little squares in here. And then we're going to go to the 10x. Brings us a little closer, and you can see the outline of one of these is one cell. So you're going to draw exactly what you see though here, and try and use color if you can. And then we're going to go up to 40x. We're going to make more light though so that we can see through. Now we can see even more detail. You can see there's one cell here right in the center. These little green dots are chloroplasts. And then this is the nucleus of the plant cell. So I do want those details drawn if you're going to move it around so that you get a really nice image of a cell. And then we're going to move on to the 100. Again, oil immersion. So we're going to add our oil and get even more detail now, and we get up close. So you're going to draw, you can only see part of a cell because we're so enlarged. All right, so we want to remove the slide. Again, we're going to clean with lens paper, put the slide away, go back to our lab sheet, and it says, label your drawings. So you want to make sure you label with, you want to label the chloroplasts and the nucleus, okay? That's some points there you don't want to miss. And now we're going to look at human blood. You're going to go through and do all the slides again, the way we just did with the plant cell. And you're going to answer your questions. And then here, synthesis and discussion. So we were comparing the blood cells to the plant cells. You want to compare their color, their shape, and their size, okay? And I made a data table for you so it would be easy for you to do that. And then you're also going to compare the bacteria to blood cells. So we would go back, and you can find the bacteria slides here when you need to look at them. You can see the blood slides are going to be in the human one. And then this page is going to have you try to identify all the different parts to see what you remember. So you can write the letters right here on the lines, and then explain what these objects do as far as the microscope is concerned. So that's the gist of the lab, and it's going to be a little time consuming because the drawing part is going to take you a while, and that's why I'm giving you 3 class periods essentially, and then all the way until Sunday to complete it. Remember though, when you're done, you must use the turn in button up here at the top right, otherwise it won't submit correctly. So you're going to turn it in using that button, and then you'll be done with the lab after you use it that way. If you have any questions, please make sure you send me a reminder, you email me, or ask if you're physically here in class. And good luck with this, and have fun with it.